Thank you for joining us for today's webinar, the first in our series of webinars about powerful collaborative research tools. Today's webinar is about how to accelerate synthetically feasible libraries from virtual screens in Mo. My name's Kim Willsher and I'm moderating today's session. We'll start with a couple of brief presentations from Marc at GridMarkets and Ben Hill at Oracle, before we hear from our guest speakers, Marc Janes and Ivan Franzoni from NuChem Sciences. We'll wrap up with a Q&A session at the end, so please send us your questions. I'm pleased to introduce our first speaker, Marc Ross. Marc is based in San Francisco and is one of the founders of GridMarkets. Before founding GridMarkets, he served as Chief Information Officer for a number of global financial services companies. Thank you, Marc, for joining us today.
Thank you, Kim. I'm going to tell you a little bit about our GridMarket service today. Our job is to accelerate computationally demanding Mo simulations and to do that at scale. We're a cloud-based molecular simulation service that not only supports Mo, but also supports Amber, NAMD, and GROMACS, and we have more to come. Regarding the Mo service, these are the key highlights. To set up, it takes 3 simple steps. You can be up and running in a matter of minutes, and then you can submit directly from the Mo user interface, or if you prefer, from a command line interface. You can then use unlimited Mo tokens regardless of how many local tokens you have, and you can also use as many machines as you want per submission. You can also stack your submissions, so you can have multiple submissions running in parallel, and the combination of those two allow you to get things done very quickly. Our model is a pay-as-you-go model, so you pay only for what you use, and it's an all-inclusive per machine hour price. So you can use that to predict in advance what the cost of your submission is going to be. And very importantly, our service is powered, it's secured by the Oracle Cloud, and you're going to hear a bit more about that from Ben, who's coming up. So how do you get started? It's very easy. You go to our website, pharma.gridmarkets.com/signup, and you create an account. There's a small form there that you fill out with some basic information, and that will establish your free account. And it will also provide you some free credits so you can trial our service. Then you download our plugins, and I'll tell you a little bit more about the plugins in just a moment. There's 2 of them. One is called Envoy, and the other, in the case of Mo, is the SVL plugin. So you download those plugins and install them, in the case of the SVL plugin, into Mo, and it looks like this. So your local instance will have both of those 2 programs installed. And then from your Mo interface, you can open up a dialog box, which you'll see in a moment, and select how many machines you want to run on. Here's that dialog box. So you can use the different parameters in this dialog box to decide, for example, how many machines you want. Here's the machine slider and various other parameters as well. So very easy. You fill out the form and you hit submit. When you do that, Envoy then is responsible for getting your project files to the CCG GridMarkets cloud, and it does that securely. And once it's there, it then spins up however many machines you wanted with the appropriate number of Mo tokens. Again, no use of your local tokens. So if you only have 3 local tokens and you need 30, we supply the 30. Or if you need 300, we supply the 300. As your job is running, then you get real-time updates via Envoy, and it looks like this. So you have this dashboard which shows the different jobs that you have submitted and the states of those jobs. You can see down here, this one, for example, is running. This one is waiting to run. You have the ability to control these jobs, to start them, to restart them, to stop them, and so on from this interface. You can also interrogate all the way down to the log level, and here's a view of what that looks like. So if you wanted to find out what logs were being produced by Mo during the submission, you could use Envoy to interrogate that as well. As your job finishes, it automatically downloads. And again, Envoy is responsible not only for uploading your files, but also downloading the results. You designated some location to put the results, and in this diagram, it's a local folder, but it could be anywhere. So when your job finishes, those results are downloaded to that folder that you designated. Let's talk about what your options are and how the GridMarkets solution compares to those options. there's basically three ways of submitting Mo simulations. One is to do them locally on your own local cluster. The other could be setting up something on AWS. And the third obviously would be the GridMarkets solution. And let's see how those compare. To set up, in the case of GridMarkets, as I said earlier, it's a very simple process. Takes you a few steps and a few minutes and you're done. Whereas setting up a local cluster is very complex, and setting up an AWS cluster is also complex, involving over 80 steps. There's the issue of scaling your tokens. If you are using a local cluster or an AWS solution, you're using your own tokens. And so as a consequence, you have a limitation on how much you can scale. In the case of GridMarkets, because of our partnership with CCG, you can scale to an unlimited number of tokens as you need them, and you only pay for them again while you're using those tokens. Again, in the case of scaling machines, obviously the AWS solution has the ability to scale on the number of machines. Your local cluster is fixed to however many machines you decided to include in that cluster. In the case of GridMarkets, you can scale to as many machines as you need. Setting up, in the case of GridMarkets, is free. It's definitely not free to set up a local cluster. Same for AWS. Maintenance, there is no maintenance for the GridMarkets cluster. The GridMarkets solution, but there is most certainly for the local cluster and to some extent for the AWS solution as well. And on a cost basis for a usage basis, the GridMarkets solution, as mentioned, is priced on a per machine hour basis, which is all inclusive. Whereas in the case of AWS, you have a bill at the end of the month, which is extremely difficult to predict. And of course, the local cluster typically will involve capital costs and other ongoing expenses to maintain and to use. So the GridMarkets solution, in short, from a usage perspective, is superior. Lastly, return on investment. The GridMarkets solution is you pay for it only as you are using it. So it has a very high return on investment as a consequence.
Our next speaker is Ben Hill, Global Director for Oracle Cloud Infrastructure. He's going to talk to us briefly about the security that Oracle brings to the table. Ben is based in Seattle, and prior to his current role, he was a product manager and cloud architect with over 15 years of cloud experience. Over to you, Ben.
Thanks again, Kim. Again, my name is Ben Hill. I'm a Global Director for Oracle Cloud Infrastructure. So when Oracle went about launching our Gen 2 cloud offering, our approach to security was deliberate and direct. We wanted to ensure that it was architected in, automated, always on, and best of all, free. So how do we go about doing this? Let's go through some of the high points. More security by default. Firstly, we designed for isolation. We've taken the hypervisor and put it on the network layer. So what on earth does that mean? Well, in short, this approach separates customer traffic, which provides better security and performance. And as an added bonus, it reduces the risk of hypervisor-based attacks. Also, OCI has what's called separation of duty, which means Oracle admins have no access to the customer's memory space. And as it relates to your data and associated storage devices, we have full encryption at rest and in motion, as well as integrated backups for business continuity and disaster recovery best practices. Now let's chat a little bit about auto detection and remediation capabilities, specifically Oracle Cloud Guard and OCI security zones. Oracle Cloud Guard provides comprehensive end-to-end monitoring for your cloud environments by continuously collecting and analyzing service configurations, audit logs, and other information, and reporting its findings as problems based on either out-of-the-box security scripts or custom security scripts created by the admin. And to complement Oracle Cloud Guard, Oracle has OCI security zones, which provide a secure enclave within a customer tenancy for the most sensitive workloads, where security is mandatory and always on. This helps ensure compute, networking, storage, and database resources comply with security principles such as always on encryption, no public access, and automated backups. And lastly, and most importantly, these services are free, like, again, multi-factor authentication for user access, OCI Vault, which can be used to store passwords, SSH keys, and certificates, and OCI welcomes third-party tools, so you can federate identity and access management policies with your organization's centralized identity provider. All right, Kim, back to you for the exciting stuff now.
Thanks a lot, Ben. Next up, it's my pleasure to introduce Dr. Marc James. Marc did his undergraduate studies in chemistry at the University of Montreal, in addition to his MSc and PhD thesis work. He's a serial entrepreneur who declined an opportunity to conduct postdoctoral research at MIT to launch his first business in life sciences about 15 years ago. Marc is passionate about improving human health through collaborative projects involving highly engaged individuals, in addition to building and growing businesses that are structured to create value in the process. He's now partner, board member, and vice president of business development and innovation at NuChem Sciences Inc. in Canada. Over to you, Marc.
Thank you, Kim. Thank you, GridMarket and Oracle, for being the driving force behind the webinar today. I want to thank the audience as well for being part of this as well. I'm happy today to be part of this event and to be one of two NuChem representatives here today. So NuChem is a small molecule drug discovery and chemical development collaborative contract research organization. And our objective at large is understanding the challenges that pharma and biotechs are facing through communications and through our process. And our promise becomes to transform ideas through science. So NuChem, at the end of the day, is a business that puts science first for results. You'll have to take our word for it today, but we do have a proven track record, an unparalleled knowledge base, and a very strong culture of innovation at NuChem. We try to provide a collaborative engagement for every project and every stage of the discovery process as well. NuChem was founded in 2011. To give you a sense of scale, we're about 125 team members in Montreal, Canada. We have about 100 team members in Quebec City in Canada as well. Out of that, I would say 85% are scientists, essentially mostly PhD scientists. And we work a lot with VC-driven biotechs in major pharma in the US, where the biotech hubs reside, the Boston area, California, the New York in development, and others. But we also work with international and worldwide customers. We offer project management, basically eliminate bandwidth issues that you might be facing. We offer services in synthetic and medicinal chemistry with the goal of identifying and solving chemistry issues that might be inherent to the projects and really drive SAR and the creation of valuable new chemical matter that is owned by our pharma and biotech partners. We offer in vitro biology and biochemistry. So the goal is to have reliable data to take our key decisions so we can develop assays for new targets, internalize assays that are already running out there, and then we can really generate meaningful data for every project because all of the analysis is based on the data itself. At the end of the day, we also need to show in vivo efficacy. So we do offer in vivo pharmacology, basically PD, PK, translational models. So we have various in vivo disease models that are key to proving a POC and translational in your projects. We also leverage third-party services. So there's two instances. So there are very commodity-based services and very specialized services. So for today's example, we do work with, for example, Chemical Computing Group and GridMarkets as specialized services. We use in silico work and support at different stages like hit finding, hit to lead optimization, but also we try to bring it into the world of chemical development to improve not only SAR and medicinal chemistry, but also synthetic transformations themselves. Computational chemistry, as you might imagine, is an integral part of the team, and it's really the key to bringing it all together from the MedChem perspective. So really examples of the things that we do is hit finding, virtual screening. So basically preparing virtual databases, preparing docking models, and running virtual screens. We also, of course, run structure-based design type studies and projects. So we generate 3D models. We do site analysis and docking studies, which support day-to-day medicinal chemistry purposes, but also validating early medicinal chemistry premises at the beginning of projects. We also do ligand-based design, so database preparation, pharmacophore elucidation, and QSAR and SAR analysis. Lastly, for me, we also do cheminformatics, database management, data handling and sharing, and data clustering and filtering. So the amount of data that's generated in project these days really need to be organized and filtered and digested for everybody to be able to use it efficiently. And that's what cheminformatics brings to the table. So that's it for me today. Thank you, everyone, again.
Thanks very much, Marc. It's now my pleasure to introduce our next speaker, Ivan Franzoni, PhD, Senior Scientist at NuChem Sciences. After completing his studies as a computational and synthetic organic chemist in Geneva and Toronto, Ivan joined NuChem Sciences, where he applies his mixed experimental and theoretical expertise in medicinal chemistry and drug discovery programs. Ivan is going to present a case study on leveraging databases of commercially available building blocks, allowing synthetically feasible libraries to be built in Mo and enabling virtual screening in the cloud using GridMarkets in the crowded kinase IP space. Over to you, Ivan.
Thank you, Kim, for the introduction, and it's a pleasure for me to be here today to share with you key aspects of a recent project we have been working at NuChem Sciences. My presentation today will focus mostly on our approach to tackle specific goals of the program and how the tools offered by the CCG Group and GridMarkets have been pivotal for the success of this study. As you may be aware, computational chemistry can have a tremendous impact in drug discovery, especially in the early stages, such as finding an initial hit molecule, its development into more lead compounds, and via an SR campaign, and the further optimization of these leads into clinical candidates. These three early drug discovery stages are more commonly referred as hit finding, hit to lead, and lead optimization. In the case study I'm going to present today, our client approached us with a program at the second stage, hit to lead. Specifically, the main goal of the project is the expansion of initial promising chemical structures identified by our medicinal chemistry team into one or more novel classes of inhibitors for the target protein, in this case, a kinase. As in many other cases, this project was characterized by specific challenges associated with the complexity of the target that can exist in multiple conformational state and the limited and almost non-existing structural information on this specific kinase target. Another particularly challenging task in the development of small molecule inhibitors for kinase receptors is the extremely congested IP spaces around these classes of compounds. Since kinases are critical in the regulation of a multitude of cellular pathways, they have been of great interest for the drug discovery community as targets for the treatment of a wide array of diseases. When we're faced with a new project, the first step is to fully understand the scientific background, specific goals, and to identify the challenges associated to the program. At the outset of a new medicinal chemistry mandate at NuChem, we work closer with our clients to gather as much information as possible from them and to fully understand their vision. Once we gather the necessary information, we then proceed to perform an independent assessment of the project to familiarize with the new target. In this case, we first needed to understand if a computational approach was suitable for the program. This was done by integration of the material obtained from the stakeholders with literature research, previous computational studies, analyze existing data for the target, assess the quality of available co-crystal structure, if any, and so forth. Alongside this initial computational assessment, our medicinal chemistry team and IP expert dive deep into all the prior art related to the target of interest to establish the best approaches for molecular design. This analysis is crucial to establish the freedom to operate in terms of chemical space, as well as to guarantee a strong IP position for all the proposed chemical entities proprietary to the project sponsor. After roughly 30 days equivalence among both computational and medicinal chemistry assessment, we're then in the position to propose a fully integrated path forward that, depending on the requirements of the project, can cover all the aspects of a drug discovery plan. This plan includes specific details regarding budget, timeline, step-by-step breakdown of the program, milestones, etc. In the project I'm going to discuss today, this parallel computational and medicinal chemistry evaluation led to the identification of 6 diverse scaffolds that fulfill all the requirements in terms of chemical feasibility, applicability to the target, as well as clear IP status. In order to help the prioritization of further synthetic effort and provide additional chemical ideas, we decided to apply computational chemistry tool in a structure-based design approach. The application of a tridimensional model of the target protein was intersected with other critical aspects of the development of novel inhibitors. Specifically, in our case, we had to work in a quite narrow window in terms of desired physicochemical properties, which include the non-trivial task to design molecules able to cross the blood-brain barrier. Furthermore, a common challenge we encounter when dealing with ubiquitous protein, selective inhibition of the target kinase is a key parameter to keep into consideration to avoid off-target toxic effect. And of course, all this while keeping in mind the requirements to ensure a good IP position for all our proposals. Based on these premises and the preliminary work done by our team, we decided to apply computational models to guide a better understanding of the binding mode and potential chemical modifications on 3 scaffolds that show the most promising features. These molecules are expected to act as orthosteric kinase inhibitors, referred as type 1 inhibitors, that binds at the same site as the endogenous substrate for each one of the 100 kinases, all the 100 known kinases, that is ATP. As of today, no public structure for this kinase are known. Our first step was to generate an homology model. Using the Mo utilities and online resources, we first analyzed the sequence of our target to identify close analogs to be considered as template for the generation of a 3D homology model. Our analysis led to identify 2 potential candidates showing 29 and 37 sequence identity with our kinase. For both examples, several crystal structures in both apo and holo states are available, but we decided to focus our attention on the template at 29% identity because although the identity is slightly lower, few co-crystal structure of this template kinase in the active state bound to a type 1 inhibitor are available. That's exactly what we need to build a model consistent with our goal, the development of a novel type 1 inhibitor targeting the active state of our kinase. So once we selected our template, we applied the tools available in Mo to first build several homology models, assess their quality in terms of geometry, and finally select one model for subsequent studies. Now that we have a working model, we next turn our attention to develop a docking protocol for the evaluation of our design proposals. This preliminary optimization was carried out in a quite standard way, where first we analyze in detail the properties of the binding site and exploring different parameters for the docking calculation using the few preliminary data that we generated in-house as benchmark data. During this preliminary analysis, we also built a pharmacophore model, including the minimum features that we identified to guarantee good activity for our compounds. As common in several type 1 kinase inhibitors, the first requirement is the binding to the Hinge domain via an H-bond donor and acceptor interactions. In the case of our kinase, an additional, not only important, but seemingly essential requirement to achieve good potency is the placement of an aromatic moiety of the ligand in a very specific position of the binding site, a feature that was easily included into our pharmacophore query. Now that we have a working homology model and a docking protocol, it's time to apply this computational tool for the generation of new proposals to be considered for synthesis. Given my background as a synthetic organic chemistry, I recognize the importance to propose compounds that can be readily prepared in the lab, especially at the beginning of a hit-to-lead SAR campaign, as it is our case here. In order to do so, it is important to incorporate the experimental synthetic sequences in any computational approach. While we were setting up the first docking protocol for this project, my colleagues were busy in the lab to design and execute the synthesis of several proof-of-concept compounds. In doing so, they were able to optimize the synthesis of the compounds of interest in a highly modular fashion with the use of simple and readily available reagents. That approach allows for the combination of different groups and fragments of the desired molecules in different orders, leading the possibility to explore the SAR of different parts of the molecules by preparing the corresponding library in a late-stage functionalization fashion. This is probably one of the best scenarios for the computational chemist as well, since it allows the generation and evaluation of virtual combinatorial libraries that is quite certain can be also prepared in the lab. Based on the versatility of the synthesis of these compounds and other analyses performed on the homology model we generated before, we focused our attention in the application of computational chemistry in the virtual screening of several potential alternatives to these three groups, indicated here by a circle, a triangle, and a square. So we began our combinatorial study by focusing on a virtual SAR on the functional group identified by the blurred circle. The first chloro intermediate shown here can be prepared in gram quantities and subsequently can be reacted with amine building blocks via either transition metal catalyzed glucoaldehyde amination or nucleophilic aromatic substitution reactions, depending, of course, on the nature of the aromatic ring in the starting material. To generate our first virtual combinatorial library, we applied the corresponding functionality in Mo. So once we drafted the generic reaction to be applied in the software, it was time to proceed with the selection of suitable and commercially available blue circle amine building blocks. For this purpose, we selected the MolPort database that contains more than 8 million compounds among regions and screening compounds. Our initial search led to the identification of a bit more than 110,000 amine building blocks that were subsequently applied in the combinatorial library in Mo. Out of the potential 140,000 possible compounds, several physiochemical descriptors were applied to filter the results, leaving a bit more than 2,000 virtual compounds to be evaluated by docking. Now, with our library of 2,000 virtual compounds and the optimized docking model for our target kinase, the only thing left to do is to proceed with the docking calculation. So these calculations were run on the GridMarkets cloud, where we took the advantage of the integration of the GridMarket plugins into the Mo interface, as well as a remarkably easy and straightforward job submission process. Although a library of 2,000 compounds for docking is not really huge per se, the use of the high computational power available on the GridMarket cloud allowed the docking study to be completed within 1 hour while leaving our Mo site license available for other tasks. Once docked, the resulting computed poses were analyzed via visual inspection to ensure a logical orientation of the ligand in the binding pocket. Compounds were selected on the basis of their docking score, tighter ranges in terms of physiochemical properties, and to prioritize simpler design at first. The analysis of the docking results led to a selection of circa 170 compounds. Of note, given the premises of this study, every single molecule among these 170 compounds is synthetically accessible and can be obtained via late-stage functionalization of a common intermediate, as discussed before. All these compounds were proposed to the NuChem and stakeholders team, and 35 were given green light for synthesis. So the first batch of 10 compounds was prepared within 2 weeks, and tested in enzymatic acid to determine their activity against the kinase of interest. And the results were perfectly in line with the expectations, and all the compounds shown a promising activity with some of the best hit at around 5 to 10 nanomolar IC50 values. In conclusion, our approach was made possible by several functionalities included in the Mo software package, as well as the easy to use and efficient computational resources provided by GridMarkets. The combination of computational chemistry with the strong interaction of, the strong integration of experimental data, synthetic chemistry approaches, and close interaction with the medicinal chemistry team allowed us to propose, design, and test several compounds, among which very active molecules were identified. The project is just at the start, but it is evident that computational chemistry had and will provide a major impact in the development of new classes of inhibitors for this specific kinase protein target. So with this, I conclude my part. So the floor is back to Kim. Thank you very much.
Thanks very much, Ivan. We now have time to take a few questions from the audience. First, we have a question for you, Ivan. In the absence of crystal structures of your target kinase, have you considered using a model predicted by AlphaFold instead of the homology model you prepared?
Absolutely, yes. So now we have these technologies available, and AlphaFold is releasing all the predicted structures publicly. So we compared our homology model with the AlphaFold kinase model for this specific kinase. So the only limitation there is that the structure provided by AlphaFold, they are in the apostate, so there is no small molecule bound to the receptor that can impact the conformation of the binding site. And also in the case of kinase, in the case of this particular kinase, the predicted state of the receptor is the inactive one, whereas we were focusing on the active state of our kinase target. But that's definitely something we consider as well. And of course, now we will consider it anytime we will be in the situation that no available crystal structure or no available structure information is it's disclosed for a specific target. AlphaFold represents for sure a very good alternative.
Great, thanks, Ivan. Next, we have a question for Marc Ross. How is the GridMarkets service provided?
Thanks, Kim. Let me show you. I'm going to share my screen, and I think a picture is worth 1,000 words. So I've gone to the pharma.gridmarkets.com/mo page, and here is the page. And if you scroll down, there's a section on pricing, and we price either by the hour, which this section describes, or by the week. It actually is less than the week. You can go down to a half-day term or a day term, and then in units of weeks. In the case of the pricing per hour, use these two sliders to estimate what your job is going to cost. So let's say, for example, you wanted to use 10 concurrent machines. I would slide this over until the concurrent machines show 10. And my estimate for the job duration might be, say, 5 hours. Let's put 5 hours in there for fun. And you can see then what the estimate of the cost is going to be. In this case, the excuse me, the per machine hour cost is $24, and the total job cost in this case for 10 machines at 5 hours is $1,215. Again, down here, if you have a number of submissions perhaps that you want to do in a given period, or you just want to, in effect, create your own cluster for a short period of time, you can use the by the week option. These columns here reserve a certain number of machines for you for that period of time, and you can reserve 5, 10, up to 60 machines for, again, a half a day, 1 day, or whatever suits you. So in short, you can pay by the hour, or you can pay by the week. Thank you, Kim.
Thanks, Marc. Ivan, we have another question for you. In the generation of the combinatorial library, you showed the process to virtually explore an SAR on the blue circle fragment. What about the other possible fragments? Is the same approach applicable there?
Absolutely, yes. So unfortunately, I could not go too much into the details because of confidentiality, but the way in which our chemists here at NuChem designed the synthetic pathways to access those compounds, it's really, it's exactly like playing with Legos. You can put them together in different ways and eventually reach the same final result. So the process I presented today, of course, is also being applied to all the other two fragments that we didn't discuss today in detail, but absolutely. And this is really pivotal because computationally speaking, you can do whatever you want, but in this case, the strength of this approach is really that can be directly matched and mirrored in the lab by the synthetic chemists.
Great, thanks, Ivan. Now we have a question for Ben. Someone in the audience says, we would be very concerned about securing our submissions. How can we be sure that this sensitive information and data will be safeguarded?
No, that's a good question. So as I had mentioned in my short presentation, all data in flight and at rest is fully encrypted by default. So there's that. And then also our admins have no access to memory space. And because of our networking configuration and how we set up a hypervisor, there's separation of compartments and separation of duties by default as well. So it's literally impossible for anybody to access files either in flight or at rest when encrypted.
Great, thanks, Ben.
Yeah.
Ivan, one more question for you. When you prepared the homology model, you decided to use the template with slightly lower identity to your target because of better availability of more relevant co-crystal structures. What about the identity between the residues in the binding pocket?
Absolutely. So that's, I didn't go too much into the details there, of every detail of the generation of the homology model. But that's, of course, a very important aspect. So not only you have to consider the sequence identity between your target protein and the receptor as a whole, so the full receptor, but also you need to consider that in the binding site, the residues are highly conserved to really be more and more confident that the binding mode that you can model in one receptor can be translated into the other one. That's absolutely something we have done in this case. So in both cases, the two templates with 29, 37% sequence identity, the identity in the pocket was something above 90% in both cases. That is a very good, it's a very good point to generate an homology model, but at the same time, it may actually provide some issues in terms of selectivity, but this is something we are working on in terms of chemical design.
Great, thanks, Ivan. We have time for just one final question. This one is for Marc Janes. Do you always include computational chemistry in your drug discovery project?
Thanks, Kim. Another good question. I'm happy to get a question. The short answer, it's a bit philosophical, depends on a lot of parameters, but the short answer is I wish we could include the computational in every project. It would be hard to imagine a project these days in medicinal chemistry with all the tools out there and the power that it brings to not include it in. And we have also something called the Discovery Partner Program at NuChem, which is sort of a way to work and think about projects where we prioritize the need for sufficient resources to ensure like successful project outcome. We don't want to get into a project that's not set up for success. So sometimes there are cases where, for budget reasons, for example, but doesn't happen that often, or there are no crystal structures of protein, but in these cases we still involve in silico support at the minimum, cheminformatics to manage data, cluster data, and organize all of that, I would say. In very early projects, we usually include a lot of computational to get a handle on things and a deep understanding. And as projects reach cruising speed, sometimes it can be anything between 10% to 40% of a modeler's time dedicated to a single projects. But in short, again, it's like we think it's an essential part of the medicinal chemistry projects, and not using it would be like navigating blindly into fog, without boundaries. So.
We hope you can join us for our next webinar on powerful collaborative research tools. NuChem will present a case study on a particularly challenging drug discovery project where cloud computing came to the rescue. This will take place on Wednesday, December 1st at 9 a.m. Pacific time. If you'd like to get in touch with NuChem Sciences, you can email them at info@nuchemsciences.com. If you'd like to contact GridMarkets, email pharma@gridmarkets.com, and to contact Oracle, visit oracle.com. Thank you very much for joining us. We hope to see you at our next webinar.